The overall goal of cargo mapping is to determine the relative amount and type of molecular motor proteins that associate with and move vesicular cargoes, and to correlate this composition of motors with the directionality of live cargo movement. This is accomplished by first growing primary mouse hippocampal neurons in microfluidic devices so that axons grow relatively straight, and by recording the live intracellular movement of fluorescently labeled cargo proteins in the axons of transiently transfected neurons. The second step is to fix neurons during the course of live cargo imaging and subsequently stain the axons with antibodies against molecular motor proteins, such as kinesins and dyneins, followed by immunofluorescence microscopy imaging. Next, the fixed cargo and motor images are aligned to the plots of cargo movement over time, also called chymographs, in order to correlate the live cargo movement with the stained associated motors as determined by the immunofluorescence staining. The final step is to determine the subpixel positions of cargo and motor proteins using a custom-developed MATLAB software package called motor colocalization. This software provides the precise XY coordinates of each cargo and motor position, as well as the relative amount of fluorescence intensities associated with cargoes and motors. Ultimately, correlation of fluorescence microscopy of live and fixed cargoes and motors at the subpixel resolution level provides information on the types of motors that are associated with specific cargoes for which the directionality of movement